case you don't know, I'm Gail Taylor. I'm one of the co-chairs for the Energy Multidisciplinary Research Group, or USRG as they're sometimes known, the University Strategic Research Groups. So what do the USRGs do, for those of you who are not aware? These are the groups that are cross-disciplinary, which are meant to encourage us to work together in multidisciplinary ways. And the reason the university initiated these groups, or one of the reasons, is that they identified that some of the big research questions in societies today require multidisciplinary thinking. And probably energy absolutely exemplifies that. There's hardly a day goes by when energy is not in the news. It was two lots of energy news today about nuclear, and also still about the cost of wind and how that might change over the coming decades. So it's an absolutely critical area of research. We have a huge amount of research in energy at the University of Southampton. We're fairly dispersed all over the place, so it's great to get together on an occasion like this. And it's particularly nice to see some of the early career researchers who are participating in the day. So what does the Energy USRG do? Well, there are 5 of us who co-chair it. Myself, Bakr, who's from Engineering and Civils. We've got Professor Paul Lewin over there at the back, for those of you who don't know who's in ECS. We've got Robert Raja here today, who's in Chemistry, and also Professor Graham Smith in Politics. So we are a multidisciplinary group of people. We meet very frequently, about once a month, and we're trying to engage with all of you and to increase activity in this area. So this year we've had today our Awayday. That's about us informing each other of what we all do, because in such a large, diverse group, it's pretty difficult sometimes to know what the guy next door is doing. So it's a way of informing each other what we do. So we thought we'd do a bit of speed dating or speed presentations. So thank you to all of you who've contributed to that. And then we also want to do some other things this year. We want to increase our external presence, so we're starting to work on our website development. And I'd ask you, if you get little emails for bits of information, please contribute those, because we do really need them to make our website dynamic. And please put the 25th of September in your diaries for a date which is going to showcase some of our work, more to the external environment. So we have Professor Jim Skiu visiting that day. And for those of you who don't know, he's the EPSRC newly appointed research champion, who's looking at what the future of RC UK energy research is likely to look like. So he'll be making the roadmaps for research. So he's a pretty important character. So that's our activity over the coming months. As some of you may know, some of the USRGs have moved towards a slightly accelerated route for development. And so the one I'm most familiar with is the Institute for Life Sciences, which of course has a nice new building, but is also a cross-faculty initiative in life sciences. And the co-chairs, the group of us, the 5 of us, are quite keen to explore whether we can take energy to that next level. We certainly have enough activity, and we're working towards that this year. And so we're keen to hear from all of you on that topic. And one thing that we think would be a good idea for Southampton, and this was an idea from Paul Lewin, is that we engage in an external review. So we invite some external experts to look at our activity and to see where our strengths and weaknesses are, and to see where we should be going in the future, and to look at the IFLS model and see if we could have some sort of institute status. So to raise the status of the USRG in some way. And as a prerequisite to that, we've posed 3 questions in the breakout group after lunch that we'd like you to think about. You might start thinking about those now. They're on the front cover here of the handout. To see if we're in good shape, and to see if there's enough energy, excitement and interest to consider accelerating our status. We're running a little bit late already, so I don't think I have anything else to say except to welcome our first chair, who is Bakr. And I hope this works with our 5-minute presentations, but he's in charge of the timing of the first session. So So our first session is in quite biological things, transport, carbon capture and sequestration. So welcome and best of luck to everyone. Back over to you.
Thank you very much. Thank you very much, Gabe. We'll try and see if we can you see this? All of you can see it? Can you see the writing? The next slide will be very small font. So I'm just testing your answers. So can I have somebody to move this table with me back a little bit to make it clearer? Okay, right. I think each presenter has got 5 minutes, and we've got somebody to tell you how many minutes left, and I've got a dog that barks at you if you don't stop. When the dog barks, it means you're out of music. Okay, let's have the first paper. It's by Tim Layton, Acoustic Detection of Undersea Gas Reserves and Leaks.
Thank you very much. Thank you for inviting me. I'm sorry I can't be with you all day. I've got another meeting in 90 minutes at Highfield, but I'll stay till then. Acoustic detection of undersea gas leaks and reserves myself. Paul White and our PhD student Benoit Burgess, who's having a poster at the back. Okay, so this is a drop of milk falling into a cup of coffee, and if you watch, it's a familiar picture. The milk goes down, forms this crown thing. But the fluid dynamics are such that substantially the same drop of milk comes out as went in. So afterwards you can play a little game, and when somebody's pouring your coffee next door and they say, do you want it white or black? You can say white. They say no, and grab that first drop and still retain black coffee. Now that makes the familiar blink noise, that's a dripping tap noise that keeps you awake at night. But interestingly, if you take a camera and film, this is the air, this is the water, so the camera lens is half submerged, there's the drop coming down, and this is an underwater microphone. This is the time series of the signal. You can see that's the plink noise that you hear, the lightly damped oscillator giving off a sinusoidal note. But the two are synchronized by a red line that moves across this film. And if you watch that synchronization, let's do it here. Here, you'll see all that makes no sound. No sound, no sound, no sound, no sound, still no sound. And the sound is going to appear just as this crater closes and pinched off a bubble from the bottom. So that tiny little bubble is making all that sound, and nothing else that's happening on the screen is making any sound. I'll run it for you again and use up another few seconds on my side, because it's nice. It's a nice thing. I'll just give you dog barks. It makes the blink. So, and that's because the bubble's like a little bell underwater. It makes a very powerful source of sound. In fact, bubble's the most powerful underwater source of sound. So if you're interested in energy and you've got a gas pipeline underneath the ocean and it begins to leak, then each bubble that's injected into water from that generates a note. And that note depends on its size. If it's a big bubble, it gives a blunk, low note. Little bubble, a blink. You know, this produces a high note, blink. It's a dripping tap noise. Whereas a scuba diver goes blah, 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 producing big bubbles, low notes. So each bubble that comes from a gas leak sings one note, and that note tells you its size. So if you listen to the chorus of sound made from a gas leak, you can say that's made by a choir of bubbles, each one singing one note. And by listening to the song, you know how many bubbles there were and what size they were, which is very powerful when you're trying to diagnose and remotely monitor a gas leak from a pipeline using an energy-efficient passive monitoring system, something that can even harvest energy from its currents. So you can think of it as a gas leak pipe detector for remote monitoring. But there's other sources of gas in the ocean. This is a sonar backscatter for methane leaks. So we have methane trapped in the seabeds, some in the form of gas, some in the form of hydrate, which is a kind of ice. If it's deep enough. And it's scattering sound, because anything that generates sound can scatter it. If you go up to a grand piano and you press on the sustaining pedal and you shout at it, the strings will ring back at you. In the same way, if you send a sonar pulse into the ocean, and here there's no bubbles, and then it sees a region of bubbles, we know these bubbles are good at making sound when they're ejected, so similarly they'll scatter sound back at you. So we can look at the methane injected using methane produced into the water column and thereby the atmosphere by looking at the sonar backscatter.
We'll move there.
Thank you. So this is a standard technology. It'll give you images of where the leaks are. What we're trying to do is look at the tuning, the notes in that backscatter, and tell us how many bubbles are to actually quantify the gas effect. And this is important because, according to this guy Dylan, who worked for the US Geological Survey, the global reserve of methane in the form of hydrate is more than twice the worldwide amount of carbon to be found in all known conventional fossil fuels on Earth, they say. So we're looking at, and of course methane is a very powerful greenhouse warming gas. So we're looking at effectively developing something like this, whereby existing equipment, which are passive in red, or active in yellow, acoustic sensors, active acoustic sensors or listening devices, can look at natural seeps, either from methane or carbon capture and storage facilities, or gas pipelines, and quantify the number of bubbles produced and the amount, and thereby quantify the gas leaks remotely. We've done a couple of tests.
Stop.
Spitsbergen and Oban. Thank you very much.
Okay, next paper. Andrew Hitchcock, which is a plug-and-play photosynthesis, understanding the cyanobacteria extracellular electrogenetic pathways. Fantastic title.
Okay, hi, I'm Andy. I'm a molecular biologist working at the National Oceanography Centre. I was Tom Bibby, Tom. Here today, he doesn't normally smile that much. But, I'm part of a big project called Plug and Play Photosynthesis, which was funded by the USRG NSF think tank. About £7 million worth of funding. We're one of 4 projects. And my small part of this project is to try and work out the photosynthetic electrogenic pathway, which I'll talk about that with more in a minute. So, photosynthetic organisms are not very energy efficient. Up to 90% of the light which they take up, they do not use for useful biology, it's wasted. And we want to know, can we harness this excess potential to drive something like biofuel synthesis, electricity generation, or production of high-value products? And a number of papers have been published recently showing that photosynthetic organisms display this light-dependent electrogenic response. That is, that when you illuminate them in a microbial fuel cell, they'll produce a current. This could be done in a carbon-neutral way, and so it can obviously be good for the environment, and you could effectively produce electricity just using light. So my project is to characterise the electron transfer pathways, how you get from water splitting in a photosystem inside the cell to electrons getting outside of the cell. I'll talk about that in 1 second. Using the model photosynthetic cyanobacteria synicocystis, you can see here. So it's genome sequenced, and it's very relatively easy to genetically manipulate. And the techniques I'll be using are generating large numbers of organisms. NOC, we're very well set up to monitor photosynthetic and respiratory physiology. We've got collaborators who are electrochemists, synthetic biologists, and we're taking proteomics and transcriptomic approaches as well. So, I guess a lot of people here won't know much about bacterial cells, but this is a very simplified version of a normal gram-negative bacterial cell. An outer membrane, periplasm, subplasm membrane, cytoplasm. And in bacteria, this response has been characterised with not photosynthetic, such as Geobacter and Shionella. Electrons from respiration can be used to produce either an anode in a fuel cell, or in nature, some sort of insoluble metal oxide to allow growth. And chelae, which are appendages that go from the cell surface. MTR, which stands for metal. I've got what it stands for. That's that. And soluble redox shuttles are well characterised in metal standardizers, well characterised in these bacteria. Cells are more complicated, so they have this inner network called thylakoid membrane system, which is where photosynthesis and the majority of respiration is occurring. They don't have MTR or soluble redox shuttles, and we have no idea if the chelae is involved. So the question I wish to answer is, how do we get electrons from photosynthesis out of the cell? So that's the question. If I just put up this relatively simplified version of photosynthesis and respiration, you can see it's quite a complicated process. So far, I've made 20 plus knockout mutants, near 25 now, and I'm in the process of screening these. We use an indicative reduction assay, and we're hoping to see a loss of this light-induced extracellular reduction. I've done a number of controls. We were slightly worried that hydrogen production could be a result of this current, because at the right potential electrochemical setup, hydrogen could dissociate at the anode, but we knocked out the hydrogenase and it still shows the activity. We're in the process of mapping the contributions of respiration and photosynthesis to this response, and I'm optimising a dye-based and electrochemical screen at the moment with collaborators in Glasgow, so hopefully we can screen a lot of mutants high throughput and start to put together what's involved in this pathway, with the eventual aim of being able to knock out competing pathways and still have the cells viable and happy, so we can get increased electricity production from cyanobacteria. And I'll finish within my time. So, anyone has any questions?
Next paper. Andreas Johannes here, which is improving the photosynthesis of synthetic efficiency of microalgae using high throughput screening and next generation sequencing. Go on there.
Hello, so my name is Andreas Johansson. I'm a PhD student. I'm based at the National Oceanography Center. I'm in the same group as Andy, so I'm working with Thomas Biddy and Mark Moore at the center, and I'm also working with Richard Edwards and Bakr Bahaj at the Institute for Life Sciences. So what we're trying to do is basically to improve the efficiency at which microalgae are converting the energy in sunlight into useful compounds that we can use to produce biofuels. We're trying to improve this process under certain conditions. So if we look at this problem in more detail, it's basically the same problem that Andy talked about. If we grow these microalgae in a large, say in a large pond, for example, for the production of biofuels, we have the problem that the surface cells, if you look at the plants here in the culture, surface cells and bottom cells, the surface cells will absorb most of the sunlight, and they will then release this excess energy as ATP plus fluorescence, which means that we lose a lot of efficiency from the overall system. And one way of trying to improve this is to modify something called the light harvesting complex. This is on a molecular level, and it's basically a antenna system. So this is the cytochrome membrane of one of the cells. If you look here, you have the photosystem in the center, and surrounding it here, you have what's called the LHC, which is the antenna that actually shuttles photons into the center. And if we can modify the size of this structure, it would mean that we could get more light down to the lower cells. So that's what we're trying to do. So how would we do this? We could either do genetic engineering, but to do this we would need to have a very good understanding of the genes that are involved in this antenna system, the genes that are controlled. And at this point we don't have all the knowledge we need to do this. The other problem we have is that the genetic tools that we need to actually do this are only available for a number of small number of species as well as some model species. So we are very limited to a few number of species out of a couple of thousand. So we use a different approach instead. We look through a large number of cells and we try to pick out the cells using a computer system. We try to pick out the cells that seem to have these characteristics from the cell. And what this also allows us to do is to work with many species. So there are thousands of different microalgae species out there. So if I'm a good candidate, we can work with it regardless if we have the tools or not. So yeah, I'm just going to tell you about the system we're using. So we're using sort of like high-throughput pipeline. What we do, we select a number of species that we're interested in. We generate a large amount of mutants using a chemical agent. Then we put these cells through something called a flow cytometer, which is basically a machine that allows us to look at a single cell at a time. And if this cell fulfills the properties that we are looking for, we can then basically select this cell, put it away so we can look at it again in the future. And when we selected these cells, we put through maybe a couple of million cells with different properties. We select a few hundred. We grow each of these maybe 100 cells in large amounts. This allows us to look at them in more detail. We make sure that they are, for example, producing more biofuels and more oil, or perhaps that they have a lower chlorophyll content, whatever we're looking for. When we've done that and we're happy with the cells, there are a number of different things we can do. We can either do like transcriptomics, which is basically next generation sequencing, where we look at all the expressed genes within the mutant and compare it to the original algae. And using data analysis, we can then try to figure out which genes have been affected, which will tell us much more about how this antenna system is working. What we also can do is also to grow these cells in larger batches, just to make sure that they are actually producing things under the same conditions that would be been used in the industry, just to make sure that they're actually producing more biofuels or whatever we're looking for. So that is what we're trying to do. I should also mention, so I'm a first year PhD student. I'm basically continuing the project of a previous postdoc, so we're really at this stage here right now, so we know that the system is working. So what we're thinking about doing now is probably to go back to the first point and do a more fine-tuned selection. So, but we have some good candidates at this point. So. Thank you.
Thank you, number 4. Robert, engineering active sites for sustainable habitats.
Okay, good morning. I'm here along with my student, Chris Hine, and what I want to do is show you briefly about what we do in chemistry in terms of engineering active sites within porous architectures, mainly to do catalytic transformations, which have a bearing on sustainability, or looking at the green aspects of chemistry which come out of it. But then go on to see how we can extend some of these ideas, some of these structures, tune the active sites within these porous architectures to actually see if we can extend these applications to energy-related ones as well. So in terms of the framework architectures, they're all porous, so they're three-dimensional frameworks, and one can place within these framework architectures active sites which you desire. So these are primarily designed from a chemicals point of view, so we can go on and make bulk chemicals in the production of nylon or polyesters, to fine chemicals which could be used in pharmaceutical industry. And our specialty really is to be able to tune the active site so that it can almost mimic the active site in an enzyme. So we could see the overlap and the partnership with people like Gale, where we can look at hybrid processes for harnessing bioenergy type applications. And in addition to the framework architecture, we have a wide range of pore diameters which one can tune, again similar to the prosthetic group in an enzyme, and one can control the selectivity in these catalytic transformations. So if you suppose you want to take glucose or fructose, as people mentioned, the biofuel application, one might be able to control the product selectivity to a high degree, which in turn increases the atom efficiency of the process. So in terms of the properties, in terms of the chemical properties, which I will briefly talk about, one can get a redox site as in an enzyme, and one can do selective oxidation reactions. Or one can replace mineral acids like sulfuric and nitric acids, which again contribute to global warming, pollution aspects. And one can design solid acid catalysts, which can do the transformation in a much more benign fashion. Or one can be clever, and you can build a mixture of these active sites. You can have a redox site and a Brunstad site within the same architecture, and one can take, for example, something like a ketone directly to make a nylon intermediate. So the first step would be an oxidation step, where the ketone is converted to an oxime, and then you can do a Beckmann arrangement with the acid site to give you nylon in a much more sustainable fashion. So Professor Layton talked about methane. So these processes intrinsically involve clean oxidants. So you can use molecular oxygen, which is plentiful and available. And one can carry out these reactions, which enhances synergy from a mechanistic point of view. And so you can come up with systems which are hybrid between biological systems and chemical systems. And that is where we are moving towards in terms of taking biological feedstock. So this was a story told to me by one of the CEOs in the US of a startup company, and he said there's so much bioethanol in the US these days that it's much cheaper than water. So he was looking at very simple examples to use our solid acid catalyst, take the bioethanol and convert it to bioethylene. And similarly, you can go to bioplastics. So very simple reactions which could make a big impact in terms of how the industry works today. So most of our projects, which I mentioned there, are funded by the chemical industry, but also we are now working in partnership with USRG and some of the Grand Challenge initiatives to look at problems which are important going forward. And I think clean energy and renewable fuels becomes something which is of paramount importance in terms of looking at the energy landscape. So this is a different type of catalytic system where rather than have a framework architecture, we can engineer an active site like a metal complex or a organometallic complex, and then you can get rid of the ligands which surround the architecture to generate a naked particle which can help in sort of looking at applications which Johnson Matthey looks like, but to replace platinum group metals with more cheaper metals. So my colleague Chris will talk about the photocatalysis and harvesting marine energy. But what I want to finish on is using these hybrid approaches. One can envisage taking glucose, fructose using a solid acid site, and then one can engineer second generation biofuels from chemical methods or chemical processes. And if you look at the biodiesel process, something which one of my other students is involved in, is to produce the fatty acid methyl ester using one type of fructose site, and use that cascade approach to take the glycerol byproduct to make propane diol, which could be used in engineering plastics. You can have more details on the poster we have there, and thank you for it.
Page number 5. Page 5. It's sustainable catalysis for renewable energy generation.
Good morning, everybody. My name is Chris. I work in the research group of Dr. Bakr Bahaj. I'm going to be talking to you a little bit about what I think is one of the most exciting projects in our group. So yeah, I'll just mention we are working, this particular project is working in collaboration with the A-Star Institute for Materials Research and Engineering, and also the recently opened Southampton Marine and Maritime Institute, so that's somewhat exciting as well. So just to develop a little bit more on this idea of these porous architectures, one of the most astounding properties of these porous architectures are the really large surface areas that these materials have. I mean, I'm sort of astounded by some of it. Some of the metal organic frameworks that we can make are up to more than 10,000 square meters per gram, so square meters, meters. And even the inorganic frameworks that we can make are hundreds of square meters per gram of material. These are like microcrystalline powders that we can make. So one thing that metal organic frameworks in particular have been picked for in the last 5 to 10 years has been hydrogen storage materials. If you can exploit and use and maximize the surface area for hydrogen absorption, you can actually store much more hydrogen than you can in a pressurized gas cylinder. So that is what's kind of been at the forefront of metal organic framework research for the past 5 to 10 years. What we want to do is further use our expertise in multifunctional architectures to look at the synergy between some of these inorganic frameworks that we can generate, and also the high surface area properties of these metal organic frameworks. By bringing these together, we can hopefully try to generate these new materials that will keep the benefits of the high surface area and activity of metal organic frameworks, whilst increasing the chemical and thermal stability of these materials. So they're a bit more suitable for use in real-life applications. For example, if it was to go into an engine, it would obviously be at quite high temperatures. And currently, hydrogen storage experiments with metal organic frameworks are done at liquid nitrogen temperatures. So that's obviously an area that we can work on quite a bit. So we want to bring these ideas together to form new materials and get some synergy out of it. But being a kind of sustainable research group, we really want to look at how can we also generate hydrogen from the photocatalytic water splitting to get hydrogen and oxygen gas from that, and then to be able to then further store that for further use. So just a couple of examples of what's been done in the literature so far one of the metal oxides that are used for photocatalysis at the moment is titanium dioxide, which rutile-4. And this has a quantum yield of about 8%, which means it takes about 8% of the incident solar energy that actually gets used to transform into useful hydrogen gas. With no further optimization, current metal organic frameworks have been shown that some of them have been shown to have quantum yields of about 3.5%. So there's definitely some potential there for using these materials for photocatalysis. So can we build on that? Can we generate new types of hybrid frameworks to get much better quantum yields and to optimize this together? We can generate different types of active sites, again, as my supervisor was saying. We can have different types of nanoparticles that we can put inside these frameworks, which can be used for photocatalysis. We can even dope some of these frameworks with organometallic type species that will be intrinsic to the framework, which can then go on to perform some of these photocatalytic transformations, whether that be water splitting or degradation of dyes or other particular properties. So there's a vast diversity of potential there with these metal organic frameworks and these inorganic frameworks, and it would be really great if we can look at some of the synergistic effects of some new materials. Yeah, thank you very much for listening.
Next paper, the presenter is not here, so we'll have to delay it until the end if we finish up. So we'll go to paper number 7. So it's Biochar, a new FP7 project, and it's going to be presented by Maud Villa.
Thank you very much. So good morning, everybody. I'm going to talk about this new project, FP7. It's a European project with different partners. And we want to see develop new technologies for carbon sequestration. And so, as most of you know, there has been this increase in gas in the atmosphere, including carbon. So we want to develop technologies to remove the carbon from the atmosphere. One way you can do that is to produce biochar. So what's biochar? It's the process called pyrolysis, where you heat biomass with a low supply of oxygen. So what you start with, you start with some biomass, including manure, organic waste, bioenergy crops, and crop residues. You put through a process of pyrolysis. You can create energy from that for transport or industry. You get bio-oil. And then at the end, you also get biochar, which is basically charcoal. And you can put it back in the soil. So the cycle is actually carbon negative. So what's the potential of biochar? There's 4 main parts of the advantage of creating biochar. You can manage the waste by by recycling material. You have mitigation of climate change by sequestering carbon in the soil. You also produce energy, and also by applying biochar in the soil, you can improve the soil for more biomass production. So this is our project, Biochar. We have several partners in Europe, including France, Germany, Italy, and the UK. We are linked with industrial companies, so we want to see if this we can put it in a large-scale industry. We also look at life cycle analysis, scenario analysis to understand how this is beneficial. there's also groups that look for short and long-term stability of the biochar. And finally, in Southampton, we mainly focus on stability in field applications. So we don't just look in a lab, we also look in fields. This is our crop in West Sussex. It's a commercial willow site. And we look at soil respiration after biochar application, also production of biomass after application, and also in differences in macroorganisms in the soil. The other part of the project is also looking at the phytotoxicity of the biochar using a plant model with Arabidopsis. So we look at the toxicity. We want to know if it's beneficial for the plants or not. So there you have biochar. You can create energy, but you can also put it back in the soil. And actually, what we notice is that there is there's an increase in biomass production. So from that, you can create more energy.
Okay, paper number 8. This is by Awayday. It's how sustainable are fossil fuels. Kai is still on his memory stick, so we'll go to number 9. Greenhouse gas balance and carbon mitigation potential to biochar crops by Zoe and Harris. It should be in the
Okay, hi everybody, my name is Zoe and I'm a PhD student working with Gale. Today I'm going to give you a short overview of my project, which involves the greenhouse gas balance and carbon mitigation potential of bioenergy crops. So this is something you guys will be all aware of. These issues apply both globally and they are UK specific. So fossil fuels are running out, we only have a limited time left, so we need to find alternative energy. We also need to reduce our greenhouse gas emissions. Obviously we're becoming more and more industrialised, especially in sort of countries which are just developing now. So we're increasing our emissions globally. We also need a more sustainable source of energy, something which is renewable and won't sort of damage our environment as much. And UK, we have a limited land capacity. Land's very expensive, we have a very large population. And compared to somewhere like the US, where there's lots of land available, here in the UK we need to be very careful. It's very precious, the land. So we need to be careful what we put on it and how we change it. So in my project, I'm going to be looking at the land use change effects of a transition from grassland to short rotation coppice willow. Short rotation coppice willow, for those of you who don't know, is a crop which is quite commonly used in the UK for bioenergy. The way it works, it's planted as cuttings, it's grown for a year, then cut, which causes it to sort of become very bushy. You then grow it for 3 years on average, and it accumulates a lot of biomass. It's then cut again, harvested, and the cycle sort of continues. So you're able to accumulate a lot of mass very quickly. And the way I'm going to be doing this is using mainly these 4 techniques. I'll just run you through how they work. So the first is Eddy Covariance. Eddy Covariance, which will be looking at the ecosystem flux. So the way it works is we have sensors which are mounted above our crop, and they're able to see the footprint of the field. So we can see the carbon which is coming out of the crops and the carbon which is going into the crops. And if we look at that over time, we can see whether we're having an emission or a sequestration of carbon. We're also looking at soil analysis. We do this by taking soil cores every quarter, and then we do a chemical analysis on this. We're able to analyse for dissolved organic carbon, nitrates and ammonium. We also have a system which is called the URGA, Infrared Gas Analyzer, that stands for, and this measures our soil CO2 flux. The URGA, along with the static soil chambers, work as an enclosure over time. So what you do is you place, with the URGA, you place the chamber onto the ground, it measures for 124 seconds, and you're able to plot your CO2 concentration accumulation over time. And what that will do is it will give us sort of a representation of what's happening in the soil. So we can see whether soil processes are more responsible for what's happening in our atmosphere, or whether it's the trees which are actually fixing the carbon into their mats. Static soil chambers, again, are a closure over time. So we place the lid on top and we take gas samples at sort of 15-minute intervals. And from that we can get CO2, nitrous oxide and methane. So we're able to get a real representation of all our fluxes from the soil. These 4 techniques are applied currently in our willow field. We also have a grassland which we're being set up at the moment. And that will we'll look at the same measures in the grassland. So what we'll be able to do is see, in the grass field we're having this much flux of CO2, in the willow we're having this much flux, and what we'll be able to do is we'll be able to compare those two to see whether it's environmentally sustainable to transfer from grassland to willow. And that sort of transition is something as well which would probably be quite representative in the UK, because you don't want to be converting land that's sort of very fertile to something like bioenergy crops when we could use that for food, because we don't want that conflict. So how is this applicable for energy? Biomass crops are the only way to convert the sun's sugar into the sun's energy into simple sugars. Thank you. It's also the potential to generate energy and sequester carbon. So this means that generating energy is a real double whammy, because it's able to sequester that carbon, so we can get 2 birds with 1 stone effectively. Short rotation coppice, as I mentioned, is very, very effective with rapidly accumulating biomass. So if we can get this short rotation coppice willow to sequester as much carbon as sort of a 50-year-old deciduous forest, it's a really great means of doing that. I'm not saying it will, but it's a means which we could try and get it to do. And also it's very low input, so it means that we have very little fertiliser and very little water. My field specifically, we don't use any fertiliser or any water is added. So it's all done the old-fashioned way by nature. And where it's needed to have fertiliser or water, these inputs are very low. So we're not having sort of an additional effect, and that's generally why crops are very good. Thank you.
Okay, point number 10 by Andy Wilson, second data analysis on travel demand. Come on, you lost 10 seconds already.
Right, how do I operate? Right.
Just use the...
So apparently my slides might be all completely screwed up in some PDF to Microsoft virus conversion. Okay. Right, so let's so now something completely different. I'm interested in the human in the loop. The pesky human doesn't do what you want to do. You want to reduce the carbon that they use. And if you're... man, this slide is totally screwed. Is it really like this all the way through?
I think possibly, yeah.
Okay, my slides are almost completely disappeared, so I just have to talk anyway. So the problem is if we're going to actually design policy interventions to change how people behave, we better be able to measure what they actually do in the first place. And it's actually really, really difficult. I'm in the Transport Research Group. We actually really don't know a lot about the details of people's travel, how much they do, what mode they do, and what the factors are in the particular choices that they make. And once you get into households with multiple people and multiple cars and multiple constraints, it becomes very, very complicated. So basically anything that we can garner in terms of detail of what people do is very, very useful. Traditional approaches, you go out there and you ask people. Surveys, self-reporting, stated preference questionnaires, standard techniques from social science. Very, very expensive to do, very limited in sample size, and also affected by the fact that people can't actually recall very accurately what they do. So, so I'm sort of interested in using that. Oh, gosh, now my slides didn't come like that. Isn't that interesting? So, all kinds. I'm interested in sort of doing clever things with secondary data, accidental data, which might give you information that fills in the gaps between these surveys with very small sample sizes. And what I'm going to talk about here is a project we had actually on MOT. This really is MOT, as in the MOT test. There's a copy of one of the pass from my car, actually, from a few years ago. This came out of a conversation at an energy programme sample. It's an accidental conversation that was not funded at the sample, but which was funded subsequently, where somebody told me, do you know what, the Department of Transport put all of the MOT tests results since 2005 online, with various things like your registration plate and home address removed, I should say. So there's a degree of anonymisation of the data. I said, really? And what we did, so I discovered what you're not supposed to do is when you put this data back together, they've left the database identifier in there, which allows you to follow cars from year to year. And in here, on your test certificate, somewhere on mine, and I'm sorry this talk is so short, an hour rather, of course, you actually have the odometer reading, or at least what the tester has put down as the odometer reading. And this means we know on a year-to-year basis, apart from when there are mistakes in the data, how far each vehicle is driven for a certain kind of data. So we can analyse that. And we got a little pilot grant from the excerpt 3 months from the energy programme, basically by the direct contact with the manager, and now we've just got a 1 million follow-on where we're waiting for the grant letter for an imminent start. And that was a wiry decision with no competition. Isn't that incredible?
You must have bribed them.
So, so here's the one side of results, because there could be many more. This is the kind of thing you can do. This is actually mileage across the bottom, normalised frequency on the vertical axis. These are distributions of miles driven per day by 2 different postcodes, one of which is Kirkcaldy in North East Scotland, which is one of the most rural places in the country, and the other of which is West London, which is one of the most urban. People drive further in Scotland, we knew that. And we also know the details of the distribution, but the point is I can disaggregate to all kinds of other variables like the date of first registration, the type of vehicle, the fuel, ... and potentially more with the data sources that we're giving you. Am I near the bomb?
There you go.
I've got loads of time left. Okay, so if I had another 2 or 3 slides, I could show you the other graphs. The interesting thing is we find things that nobody knew about before. It's no surprise in these distributions that London is left shifted. People drive less in urban areas. These are for vehicles that were first registered in 2004. As you go into older and older vehicles, the difference in the mileage distributions disappears. Nobody knew this before. But now in East Scotland, the old vehicles are being driven exactly the same amount as old vehicles in London. Why? We don't know. Now, is this somehow displacement of miles within households? Your old vehicle is the second vehicle which only gets used for discretionary journeys. But it poses all kinds of interesting questions, which you could then fill in with survey data. Any questions?
One question for Eddy. He's got 1 minute to go.
Yeah. Are there any social scientists here?
No.
Yes, when we do human-computer interaction, it's really interesting. I like the chat. Did you think about...
Well, I'm sitting next to you, so
yeah, that's cool. Crowdsourcing.
Who is the polluter, right? That's the question here, right?
Crowdsourcing mobile phones, mixing with that stuff.
Yeah, we're also looking at that, and Tom, who's coming after me, is going to talk a little bit about that too. Okay.
Okay, thanks very much, Eddy.
Good morning, everyone. My name is Tom Sherratt. I'm a colleague of Eddy's in the Transport Research Group in Faculty of Engineering and Environment. So this is a project that came out of the same sandpit that Eddy was in, providing new visualizations of transport options using located media, social networking and tagging technologies. So it's called 6 Sense Transport. So what we're trying to do here really is to give people better visualizations of more collaborative transport options. So with your mobile phone knowing where you are, can we use that with the Internet of Things, tagging other things in the environment, which could also give us information, and social networking to give us better visualizations of how we might link up with other things in space and time, hopefully. So the idea is you've got social networking, so people maybe in a Facebook group who are willing to share information about things with other people, and also potentially you have smartphones so you know where they are. And then we've also got people you want some information about their schedules, what we classically do every day, our fixed habitual behaviours. So armed with this information and information about things in the environment which might be tagged, your car, where you're going, as Eddy's saying, various data on that. As I'll give you a little example in a minute, you might have other elements in the environment that have been tagged with information. You might be able to then link up with things or visualise when things might be happening in the future. So when might Tom be travelling with his kids to school? Will I be able to link up with them, and so that you can share travel options? That's the kind of thing we're looking at here. So a lot of it's about actually predicting when people might be in certain places, which you can do knowing a bit about their habitual behaviour, and also about what activities are happening in the environment through tagging various bits of technology. So we have 3 domains that we're working on in this project. I'm just going to give you a little bit of flavour about one of them, which is my area of interest, which is logistics. So we're working with Oxfam. So Oxfam is one of the biggest charities in the UK. They spend over £7 million driving our old jumpers, pants and clothes around each year, which they've rejected from shops. So stuff that you donate, which they go, God, we can't sell this, there's stains on this. It then has to be got rid of. And unlike other charities, they don't get rid of it at the back door. They have a centralised take-back system. It goes up to Huddersfield, and then it's shipped out to the Far East. Okay, so it's a lot of it's quite inefficient. And they've got over 700 shops who are all sorting stuff out. And they've got 1,500 clothing banks, so you can go to a big metal bin and put your clothes in, and that will be collected. So the idea here is you can give visibility to the shop manager, the drivers, the area manager on what's happening in the network. They haven't got any of this at the moment. So simply by giving the shop manager visibility of where vehicles are, so simple tracking of where vehicles are because the guy's got a phone that's on. But also we're working with a company called Smart Bin, so we've actually tagged a great load of their clothing bags, so we know what the fill rate is. But the interesting thing is not how full it is now. I want to know how full it is, when is it going to be full, so I can go and collect from it. So the idea is, using this type of technology, you could slide forward into the future to see when is that bank actually going to be full, so when should I go and collect from it. And at that time, because I know the schedules of the vehicle, the collections, who might be the nearest person to that bank to enable me to collect from it. So it's what we call human-centric scheduling. You're giving more power to the individuals, the shop managers, because they also use volunteers. So people volunteering in Oxfam shops will go with their own car and empty a nearby clothing bag to bring the stock back when they need more stock. So it's a very complex system. So the idea here is we're kind of giving various players within the organisation more visibility of what's happening now, but what's going to potentially be available in the future. So people might be able to link up in a better space and time to manage the operation more effectively. So that's the kind of concept. The interesting thing we found out, we've tagged a load of these bins already, and the frightening thing is that in some areas, 60% of the stuff's getting nicked. So people going around going to throw them, I'll have a bit of that piece, because second-hand recycling is worth such a lot of money now. So we get these lovely trays of these bins filling up, and you're predicting when it's going to be full, and suddenly you go, where's it all gone? And there's no driver there. And the thieves have actually gone out and put their own locks on the clothing bag. They've taken Oxfams off, they've put their own on, so the poor Oxfam driver's trying to get in. And they can't because someone's put their own lock on. So you're able to see and potentially predict when the thieves might hit a bank. So we're trying to save carbon, reduce transport by giving people more visibility of what's happening. But in actual fact, we might do the opposite, because you might have to go out to these things more often to get some value out of the bank. So you're then going, right, who is in the area who could pick up from this bank? It's only 30% full, but we've got to get some value out of it. So the area manager might be able to do it, a local volunteer might be able to do it, or one of the big centralised vehicles might be able to do it. So what we initially set out as a project to look at reducing travel, because if you know how full the bank's going to be, then therefore you don't have to go out so often. We might have to go out even more just to get some value out of it. So it's a very interesting project for me. So that's where we are. Thank you very much.
We got the stick. So one more paper before we break for coffee. And we also have another one, which is paper number 6, which Steve Gibrella apparently had to go to another meeting. So we'll have that in the next session. Kai is going to start his presentation in a minute. It's all about how sustainable are fossil fuels.
Very short presentation. It's going down.
Okay, my point is slightly general. As all of you are aware, the UK government has recently gone for the nuclear and gas. However, the nuclear part has been running into problems, and looks like we're going to rely on the fossils for a long time to come. This is current worldwide figure on the UK dependence on fossil fuels are actually greater on this. What about, so the question is that, how much reserves do we have? We have 46, 59, 118 years for oil, gas, coal. That's on current proven reserves and also the production rates. However, I have to mention that these figures have stayed more or less constant for the last 30 years. That means every year some new reserves being discovered. There are also unconventional fuels, shale gas, shale sands, oil shale, which are several times larger in reserve than the conventional fossil fuels. So we are not actually running out our fossil fuels. Based on the BP's World Energy Outlook, 2030, the world actually will will use more fossil fuels rather than reduce it. So oil, coal, gas will increase 15, 23, and 50%. So despite all the growing use of renewable energies, we still use more fossil fuels in 2030 than we are using now. Okay. So the growing use of renewable energy will help to meet the growing energy demand, but not reduce our dependence on the fossil fuels. Which ones? Which is to move forward?
The same one you did.
Okay. So what are the currently leading technologies to use fossil fuels? There's not a complete list, but some of the technologies are really promising. For example, the ultrasonic thermal power plants will increase your thermal efficiency by 30 or up to 40%. We also have the integrated gasification, combined cycle, again increase the efficiency considerably. But one of the technologies that can actually make use vast amount of coal reserve is the gasification of power generation. This technology will generate not only fuels but also heating, electricity, and chemicals at the same time. Of course, there are some new technologies in the combustion area which will actually combine the carbon capture and storage. And some of these use oxygen on the air to burn the fuel, and also the mild combustion concept, which burns fuels at very low temperature so that we can actually reduce the emissions. So some of the current projects we are running now are listed here, just within a small group, but there are some other people in our different sectors. This is what I just mentioned. Currently, the energy efficiency for power generation, fossil fuels, about 30%. This is the efficiency that most of the UK power plants have now. Okay, but the current leading technologies give you about 45, 43, 45 in that range. So by that, we can reduce almost 35% of CO2s just purely due to increasing efficiency. If we can combine the carbon capture and storage, then we obviously are on track in many ways to meet the carbon efficiency. Okay, that's all.
Thank you very much.